Welcome to the Art Bio Lab channel. Today, we're going to talk about the MTT assay. Now, this is a really fundamental test you'll find in labs all over the world, but it's also, honestly, a super clever piece of biochemical detective work. We're going to break down exactly what it is, how it works, and why it's become such an essential tool for scientists.
So let's start with a really basic question. Just imagine you're a scientist, and you've got this dish with thousands and thousands of tiny little cells. Some are healthy and happy, some might be dying, and some are already gone. How in the world do you tell the difference? I mean, you can't exactly ask them how they're feeling, right? Well, this is the exact problem the MTT assay was invented to solve.
Here's the plan for our explainer. First, we'll cover what the MTT assay actually is. Then we'll get into the, magic color change that makes the whole thing work. After that, we'll walk through the actual 6-step lab protocol, learn how to read the results, and finally, we'll look at it for what it is, a tool in the scientist toolkit with its own pros and cons.
All right, let's jump right into section 1. We'll start with the basics. What is this test, really? And what is it actually measuring?
Okay, so at its heart, the MTT assay is what's called a colorimetric method. That's just a fancy science way of saying it measures things using color. And here's the really important part. It doesn't directly count living cells. Nope. Instead, it measures their metabolic activity. You know, how busy they are, how much energy they're producing. And it turns out this activity is a fantastic stand-in for cell viability, their ability to grow, and how they react to toxins.
The whole principle is just beautifully simple. You add a yellow chemical to your cells. If those cells are alive and their metabolism is humming along, they perform a little biochemical magic and turn that yellow dye into a deep purple. But if the cells are dead or dying, nothing happens. The solution just stays yellow. It's a dead simple visual cue that's incredibly powerful.
And because it's so simple, it has huge applications. I mean, scientists use this all the time to see if a new cancer drug is actually killing tumor cells. They use it to measure how quickly cells are multiplying or to figure out how cells react to things like pollution or other environmental stressors. It's just a fundamental way to compare the basic health of cells in countless different experiments.
Okay, so let's get into our second section. We know that living cells turn this dye purple, but how? How do they do it? Well, it's not magic, it's pure biochemistry. So let's pop the hood and see what's going on.
First up, we have our main ingredient, MTT. This is that yellow tetrazolium salt we keep talking about. By itself, it's really just a yellow powder that gets dissolved into a solution. The only thing you need to remember is that it's basically just waiting around for a specific enzyme inside a living cell to come along and transform it. And this is the end product of that transformation, formazan. When the MTT gets processed by the cell, it turns into these tiny, insoluble purple crystals. Now, insoluble just means they don't dissolve in water, which is actually a really important detail that we'll come back to. So you go from a yellow dye that dissolves easily to a purple crystal that doesn't. Now here's what's really cool, where this all happens. It happens inside the mitochondria, the powerhouses of the cell. There are specific enzymes in there that are crucial for generating energy, and these are the enzymes that drive the reaction. So this means the test isn't just checking if a cell is there. It's actually checking the health of its power supply. A cell with busted mitochondria isn't going to make that purple color. Okay, so we've covered the what and the why. Now let's get practical. How does a scientist actually do an MTT assay in the lab? This section is all about the step-by-step protocol.
The whole thing is usually done in a plate with 96 little wells. First, you seed your cells into the wells. Second, you treat them with whatever you're testing, a drug, a toxin, whatever. Third, you add that yellow MTT solution. Fourth, you wait. You give the cells a few hours to do their work and make those purple formazan crystals. Now, step 5 is key. You add a solvent to dissolve those insoluble crystals, which releases all that beautiful purple color. And finally, step 6. You pop the plate into a machine that reads the absorbance. It measures exactly how purple each well is.
And that measurement brings us to our fourth section. We've got a number that represents the color in each well. But how do we turn that into something meaningful? How do we turn color into actual data?
Well, it's actually super straightforward. The machine gives you a number called absorbance, which is just a measure of how intense the purple color is. You just take the absorbance from your treated cells, divide it by the absorbance from your control cells, that's a group of perfectly healthy, untreated cells, and then you multiply by 100, and voila, that's your percent cell viability.
This bar chart shows a perfect example. See on the left? Those are our control cells, the untreated ones.
They set our baseline at 100% viability. Now look to the right. These are the cells that we treated with the drug, and they're only showing 50% viability. The conclusion is pretty clear. At that dose, the drug knocked out about half the cells. It's a clean, quantifiable result that all started from a simple color change.
So this assay sounds pretty great, right? And it is. But, like any tool, it's got its strengths and its weaknesses. So for our last section, let's take a really balanced look at the pros and the cons.
The advantages are obvious. It's simple, it's cheap, it's sensitive, and it gives you reproducible results, which is exactly why it's so popular. But there are some limitations to keep in mind. That extra step to dissolve the formazan crystals can sometimes be a bit tricky. And if the compound you're testing has its own color, well, that can interfere with the final reading. Plus, the results can sometimes be a little different depending on the type of cell you're using. It's a fantastic tool, but you have to use it carefully.
But despite those limitations, here's the bottom line. The MTT assay is an absolute workhorse in labs all across the globe. That combination of being reliable and just so easy to use means it's still a fundamental method for answering really critical questions in biology and medicine. It's really a testament to the power of a clever, simple idea.
So we'll leave you with a question to chew on. The MTT assay is powerful, but it's not perfect, right? It measures metabolic activity, not necessarily life itself. And it has a few tricky steps. So if you were going to invent the next perfect cell viability test, what would you improve? What would that test look like? It's a question that scientists are literally working on every single day.